Imagine a world ruled by giants. Massive creatures roamed the earth, their presence shaping the very landscapes they inhabited. Among these titans was the dire wolf, a predator that stalked the plains and forests of North America until its extinction around 12,000 years ago. Larger and more powerfully built than its gray wolf cousin, the dire wolf was a formidable hunter, capable of bringing down prey much larger than itself. For millennia, the dire wolf remained a creature of legend, its bones whispering tales of a bygone era. But now, thanks to groundbreaking advancements in genetics and the tireless efforts of scientists, the dire wolf is no longer a relic of the past. It has been brought back from extinction in a feat of scientific mastery that has captivated the world. The successful de-extinction of the dire wolf marks a pivotal moment in human history. It signifies our growing ability to not only understand but also manipulate the building blocks of life itself. This achievement opens up a Pandora's box of possibilities, raising profound questions about our responsibility to the natural world and the ethical implications of wielding such power. This is the story of how the dire wolf, once lost to time, has returned to reclaim its place in the tapestry of life. It is a testament to human ingenuity and a beacon of hope for the future of conservation. At the forefront of this scientific revolution is Colossal Biosciences, a company that has taken on the mantle of de-extinction. This ambitious endeavor aims to bring back species that have long vanished from our planet. Co-founded by renowned geneticist George Church and tech entrepreneur Ben Lamm, Colossal is driven by a bold vision. Their ultimate goal is to see the woolly mammoth thunder across the Arctic tundra once more, restoring a piece of our planet's lost history. However, the company's ambitions extend far beyond the woolly mammoth. They envision a future where a variety of extinct species can be brought back to life. Colossal aims to develop and refine the tools and techniques of de-extinction, pushing the boundaries of what is scientifically possible. By applying these techniques to a wide range of extinct species, they hope to revolutionize our understanding of genetics and biodiversity. The dire wolf, with its close genetic relationship to the gray wolf, presented an ideal candidate for Colossal's first foray into de-extinction. This ancient predator once roamed North America, and its return could offer valuable insights into the past. The company assembled a team of leading geneticists, paleontologists, and conservationists, all united by a shared passion for pushing the boundaries of science and exploring new frontiers. These passionate scientists worked tirelessly, driven by the excitement of discovery and the potential to make history. Their mission, to bring the dire wolf back from the brink of oblivion, to see it roam the wilds once more. This ambitious undertaking required not only cutting-edge technology, but also a deep understanding of genetics, animal husbandry, and the delicate balance of ecosystems. The team must consider the ecological impact of reintroducing an extinct species. The de-extinction process is a complex and multifaceted endeavor involving numerous scientific disciplines and meticulous planning, but it can be broken down into several key stages. Each stage is critical and requires precision and expertise. First, scientists must obtain viable DNA samples from well-preserved remains of the extinct species. This step is crucial as the quality of the DNA can significantly impact the success of the project. Next, they must analyze and sequence the ancient DNA, comparing it to the genomes of closely related living species. This comparison helps to fill in the gaps and reconstruct the extinct species' genome. This comparison allows scientists to identify the genetic differences that distinguish the extinct species from its living relatives. Understanding these differences is key to accurately recreating the extinct species. Finally, they must use sophisticated gene editing techniques to edit the genome of a living species, introducing the genetic traits of the extinct species. This step often involves the use of advanced tools like CRISPR to precisely modify the DNA. The edited cells are then cultivated and monitored closely to ensure the desired traits are expressed. This meticulous process is repeated until a viable embryo is created. The embryo is then implanted into a surrogate mother, who will carry it to term. Once born, the young animal is carefully nurtured and studied to ensure it thrives in its new environment. The ultimate goal is to reintroduce these species into the wild, where they can contribute to the ecosystem and help restore natural balances that were disrupted by their extinction. Through their groundbreaking work, Colossal Biosciences is not just resurrecting the giants of the past, but also paving the way for a future where the boundaries of science and nature are continually expanded. The journey to resurrect the dire wolf began with a treasure trove of genetic information locked away in ancient bones. These bones, remnants of a time when dire wolves roamed the earth, held the key to unlocking the secrets of this majestic predator. Over millennia, the remains of countless dire wolves had been preserved in the Brea Tar Pits, a natural asphalt deposit in Los Angeles, California. This site, rich in prehistoric fossils, has been a goldmine for paleontologists and geneticists alike. These tar pits, a time capsule of prehistoric life, offered scientists an unprecedented opportunity to peer into the genetic makeup of this long-extinct predator. The preservation conditions in the tar pits were ideal, keeping the bones in a state that allowed for the extraction of ancient DNA. Extracting DNA from ancient bones is a delicate and painstaking process. Each step requires meticulous attention to detail to avoid contamination and ensure the integrity of the genetic material. Over time, DNA degrades and becomes fragmented, making it challenging to piece together a complete genome. The older the sample, the more fragmented the DNA, which adds layers of complexity to the sequencing process. However, thanks to advancements in DNA sequencing technology, scientists at Colossal were able to overcome these obstacles and successfully extract and sequence the dire wolf genome from the Brea specimens. These technological advancements have revolutionized the field of genetics, making it possible to read and interpret ancient DNA with unprecedented accuracy. The dire wolf genome, a blueprint of life from a bygone era, revealed fascinating insights into the evolutionary history of this iconic predator. By analyzing the genome, scientists could trace the lineage and evolutionary adaptations that made the dire wolf such a formidable hunter. It confirmed that the dire wolf was a distinct species from the gray wolf, diverging from a common ancestor over 5 million years ago. This divergence marked a significant evolutionary split, leading to the development of unique traits in the dire wolf. The genome also shed light on the dire wolf's physical characteristics, such as its massive size and powerful bite. These traits were crucial for its survival and dominance as a top predator during its time. With the dire wolf genome in hand, scientists could now compare it to the genome of its closest living relative, the gray wolf. This comparison provided a deeper understanding of the genetic differences and similarities between the two species. This comparison was crucial for identifying the key genetic differences between the two species, highlighting the specific genes responsible for the dire wolf's unique traits. By pinpointing these genetic markers, scientists could better understand the evolutionary pressures that shaped the dire wolf. Armed with this knowledge, scientists could begin the next stage of the de-extinction process, using gene editing to rewrite the gray wolf genome. This groundbreaking work involves using CRISPR technology to insert the dire wolf's unique genetic traits into the gray wolf's DNA, potentially bringing the dire wolf back to life in a new form. The implications of this research extend beyond just the dire wolf, offering insights into the possibilities and ethical considerations of de-extinction and genetic engineering. The next stage of the de-extinction process involved harnessing the power of gene editing, a revolutionary technology that allows scientists to make precise changes to the DNA of living organisms. This technology has opened up new frontiers in genetic research, enabling scientists to explore possibilities that were once confined to the realm of science fiction. Using a technique called CRISPR-Cas9, scientists at Colossal were able to edit the genome of gray wolf cells, introducing the specific genes responsible for the dire wolf's unique traits. CRISPR-Cas9, which stands for Clustered Regularly Interspaced Short Palindromic Repeats and CRISPR-associated Protein 9, has revolutionized the field of genetics by providing a method to alter DNA with unprecedented accuracy. CRISPR-Cas9 acts like a pair of molecular scissors, allowing scientists to cut and paste specific DNA sequences with remarkable precision. This technique involves using a guide RNA to locate the exact spot in the DNA sequence that needs to be edited, and then the Cas9 protein makes a cut at that location. Once the DNA is cut, scientists can add or remove genetic material, effectively rewriting the genetic instructions. By introducing dire wolf genes into gray wolf cells, scientists could essentially rewrite the genetic code of the gray wolf, imbuing it with the characteristics of its extinct relative. This process is meticulous and requires a deep understanding of both the gray wolf and dire wolf genomes. Scientists must identify which genes are responsible for the dire wolf's distinct traits, such as its size, fur texture, and behavioral patterns, and then carefully integrate these genes into the gray wolf's DNA. This process is akin to taking a blueprint of a modern house and modifying it to resemble the architectural style of a bygone era. Just as an architect might add Victorian design elements to a contemporary home, scientists can incorporate ancient genes into a modern genome, creating a blend of old and new. Just as an architect can incorporate elements of Victorian design into a contemporary home, scientists can introduce ancient genes into a modern genome, effectively resurrecting traits that have been lost to time. This blending of genetic material allows for the creation of a new organism that carries the essence of its ancient predecessor while being adapted to the modern world. The goal was not to create an exact replica of the extinct dire wolf. Instead, scientists aimed to produce a proxy species, a living animal that closely resembled the dire wolf in both appearance and behavior. This approach acknowledges the limitations of current technology and the complexities of genetic engineering. Instead, scientists aimed to produce a proxy species, a living animal that closely resembled the dire wolf in both appearance and behavior. This proxy would serve as a living testament to the dire wolf's legacy, allowing researchers to study its traits and behaviors in a way that was previously impossible. This approach acknowledges that true de-extinction, in the sense of creating an exact genetic copy of an extinct species, is not currently possible. The ethical and technical challenges are immense, and the goal is to create a functional and ecological equivalent rather than a perfect clone. However, by combining the best of modern genetics with the preserved legacy of ancient DNA, scientists can bring back echoes of the past, creating creatures that embody the spirit of their extinct counterparts. This endeavor not only advances our understanding of genetics, but also offers a glimpse into the past, allowing us to appreciate the diversity of life